Hello, everybody, and welcome to today's webcast on New Horizons in Lipid Nanoparticle Production. I'm Tim Wright, editor of Contract Pharma, and today's webcast is being sponsored by Micropore Technologies. Their expertise is in the formulation of drug product delivery systems. In pharmaceuticals and biopharmaceuticals, this covers the full range from relatively simple transdermal emulsion systems to the most complex and demanding micro and nanoparticle injectables. Check them out at www.microporetech.com. We have two speakers on the line today. Dr. Yvonne Perrie is a professor at Strathclyde Institute of Pharmacy and Biomedical Sciences at the University of Strathclyde in Glasgow. Dr. Perrie is an internationally recognized expert in the field of liposomes and particulate drug delivery research. She has approximately 130 peer-reviewed manuscripts, 5 textbooks, and her work has been translated into 6 patents. Also on the line is Sam Trotter. He's the operations manager at Micropore Technologies. Based in Wilton in the UK, Sam has played a key role as a process development engineer and operations manager, working on optimizing customer formulations and equipment design for many sectors, including cosmetics, food, and pharmaceuticals. And today we're going to learn about some new technology that can be used for the anticipated major growth in lipid nanoparticle technology as a delivery system for treatment of infectious diseases and cell and gene therapies for hereditary diseases. We'll be accepting questions throughout the talk. You can send those in using the box at the bottom of your screen, and questions will be answered after the presentation in a brief Q&A. This session will be archived on our website, contractpharma.com, for 1 year. And if you're having any tech difficulties, you can type those into the same question box, and someone on the back end here will help you out. At this point, I will turn it over to Yvonne.
Thanks, and thanks for the kind introduction. So yeah, I'll talk us initially through the work we've been doing with the system and then on the data we've got so far. So just to give you a bit of background on where we come from with nucleic acid vaccines. So it was back in the 90s where Wolf et al. Showed that if you injected plasmid DNA into the muscle, you could actually get expression, and if that plasmid encoded antigen, you could actually get an immune response. And that's where DNA vaccines originally stem from. And then going forward, we've further manipulated this and enhanced it. And rather than injecting the DNA, we can go straight to the mRNA and then use the mRNA to encode a protein or an antigen, as we're seeing with COVID-19, and that will get us antibody responses. Now, the good thing with going with mRNA rather than DNA, well, there's one less barrier for you to get through as well. So mRNA is translated in the cytoplasm, whereas DNA has to get right into the nucleus. So that's a second advantage of using mRNA over DNA. So we all are very familiar now with the use of this technology in vaccines. But what we do need to remember is that mRNA vaccines do need a delivery system. They are highly negatively charged and highly water soluble, so this makes it difficult for them to get through the cell membrane. Also, they're easily and quickly broken down by enzymes, so when we handle them in the lab, we have to take extra precautions because they are degraded. So all these factors combined present barriers, and to overcome this, we use delivery systems. A range of delivery systems have been explored, so people have looked at liposomes, lipid nanoparticles, which we are seeing used in the vaccines currently, and then the variations that we have solid lipid nanoparticles, polymeric-based nanoparticles, and also cationic emulsions. And they've all been explored for the delivery of that. And actually, the use of lipid nanoparticles, or LNPs, has actually been approved previously. So in 2018, there was Onpattro was approved. So this is an ionizable lipid nanoparticle, which was approved, as I say, in 2018, and includes a therapeutic siRNA for the treatment of various diseases, as seen here, hereditary transfusion-mediated amyloidosis. Not easy for me to say, I have to admit. So that's why we need a delivery system for these mRNA systems. So in terms of what the LNP is built from, so we have two diagrams here. So on the left is a cryo-EM of a lipid nanoparticle, and you can see a more condensed core, where on the right is a liposome, and you can see that's got a more aqueous core. And if you look at the schematic representation, you can see that with an LNP, The inside is basically condensed round. So we have ionizable lipids, so they are cationic when we make the LNPs, and they bind to the negatively charged RNA. And then this pulls down the condensed core. And then also part of the lipid nanoparticles are stabilizing lipids. So we have disteryl-phosphatidylcholine, or DSPC. We have cholesterol. Both of those help stabilize and hold the particle together. We also have a pegylated lipid which presents mainly on the surface, again helping to stabilize the process. So in terms of lipids and how they've been developed, so the story really goes back to cationic lipids first being used, again for the DNA vaccines. So we had a cationic lipid, for example, as shown on the slide, DOTAP, and then that's gone through evolution. So moving away from something that's always cationic to something that we can make cationic at a lower pH. So if it's a pKa around about 6.7, if we go below that 2 units, it's going to be cationic at low pH. Then we bring the pH up when we've made the LNPs so the outside is neutral. And that reduces toxic effects which are sometimes associated with cationic lipids. So you can see that evolution from the DOTAP lipid to the D-lin KC2 DMA to the D-lin MC3 DMA, which is one of the products used in Onpattro that we were just talking about. And then you can see the stabilizing lipids. As I say, you can see disteryl-phosphatidylcholine with that long carbon chain helping to stabilize it. Cholesterol also used. So those were both previously approved in liposome products. And then we have the PEG to stabilize it. Also, some people have looked at fusogenic lipids, as shown diolylphosphatidylethylamine, DOPE. So those that give you an idea of the lipids that are being explored in these products. So we know there is 2 currently approved LNP COVID vaccines. So we have the Pfizer one, and these are the UK dates. So the Pfizer vaccine, the first global approval was actually in the 2nd of December 2020 by the MHRA, and then quickly followed on by Moderna that was approved in the 8th of January. So both of these have the stabilizing lipids, DSPC and cholesterol, so both use that, a slightly different ratio, but very similar. And then in terms of ionizable lipids, they do use 2 different ionizable lipids, so you can see here, but both of them have a pKa round about 6.5, so if we reduce a pH, they're cationic, bring the pH up, they're neutral. And both of them use a pegylated lipid, again, fairly similar, but slight changes in that. But overall, the structure is very similar, and there's just small differences in the choice of lipids used. Now, the key thing, as I said, is this effect of the ionizable lipids. So generally, in terms of production of these, we would dissolve our lipids in an ethanol phase, and then we dissolve our RNA in a buffer phase. So lipids dissolve in ethanol, RNA in the buffer, and we bring them together in a mixing process. And the buffer, we tend to start at a lower pH, pH 4.4 to 5. So that means your ionizable lipids will be in the cationic form. Then as we increase our polarity, our lipids will come out of solution and form the complexes. So they'll form LNPs. And then once they're formed with this complex, we can raise the pH up to around 7.4 so that they are neutral and can be easily injected, IM, as we see with the current vaccine. So the key thing is this mixing stage, so pushing the particles so that they actually form. And basically this forming process is basically due to us reducing the ethanol content, and the rate we reduce it controls the size of the LNPs. So if we take that ethanol concentration down quickly, we get very fast mixing and we get small particles. If we take it down slower, then the lipids have more time to form these discs, as is shown on the slide, and then they curve round and form slightly larger LNPs. So basically the point is that when we control this mixing and the rate of mixing between our ethanol and our aqueous phase, we control the size of our lipid nanoparticles. So the key is this controlling of the mixing stage when we form LNPs and having a robust and refined process for controlling that process. The aim of our research was to actually look at how we can improve the manufacturing process for RNA-based LNP systems. So we know they work well. We've seen it in the COVID crisis, how well these have performed. We know the rollout has been excellent, but as always, it should be much faster if we want to be able to promote these and get these out to a global market. So we want to be able to facilitate faster and cheaper global deployment of high throughput manufacturing methods. So we want to be able to get these LMPs out there as quickly as possible. We have been investigating the crossflow membrane micromixing system that has been developed, and we'll hear a bit more about the actual process in that. So as I say, I'll talk about how we've used the system, and we've been looking at that to develop LMPs. So our objectives were basically threefold. We wanted to be able to manufacture LMPs using the new cross-flow membrane process, wanted to investigate the impact of the production speeds on these, because obviously the faster you can go, the easier it is to manufacture at a high throughput. We also wanted to look at how the configuration parameters impacted on the LMP attributes. So basically, can we develop process control through this system and how the various processes impact on it. So in terms of lipids we used, so we used the standard format with the DSPC and cholesterol. We actually used just a cationic lipid at this stage due to supply chain at the time, and we used the standard pegylated lipid that you see used in the Moderna LMP vaccine formulation. So we used poly A, so that's basically a surrogate of RNA. So rather than encoding anything specific, it just had a long sequence of poly A in that respect. So that's exactly the same as normal mRNA in that respect. So we had a ratio of basically it's positive to negative at 6, which is comparable with the vaccines currently available and being used. So in terms of the setup, so we were using the AXF stainless steel cross-flow membrane micromixing unit. So as you can see, it's a membrane that is tubular, and we'll hear more about this in a minute, but we use the 300 micron thick with the 10 micron pore spaces and a square grid that are 200 microns apart. The advantage of this, it works at low pressure, and we achieve laminar flow of fluids, and it's all process controlled. So we looked at production speeds between 75 and 500 mils per minute. So this was installed in our lab. It was posted up to us, and it was remote installed for us. Basically, we had Zoom training sessions, so it was very easy for us to get going with it. So we didn't need to worry about process engineers coming into the facilities while we were in lockdown. So that was great to achieve that. So the key thing when you're manufacturing is the 3 elements that every regulator will look for is quality, safety and efficacy. Those are the 3 guiding principles of approval for any vaccine and any medical product. When we consider LMPs in terms of quality, a lot of the key parameters that people are looking at are particle size, polydispersity of the particle size, beta potential as long as it's round about neutral at the end, and obviously RNA loading. You need to get your payload into the formulation. So that's what were key screening attributes. So in terms of the production, we wanted to see how fast we could produce. We wanted to check the robustness of the process. We wanted to be able to have in-process control. And we wanted a decent yield. We didn't want to lose all our lipid or particularly our mRNA when it comes to it, because that's highly expensive. In terms of characterization, we did particle size and polydispersity using the Malvern system, which is similar across the world. Most people are using that. And we used a ribal green assay to measure RNA condensate. So if we look first, here is where we are. So first things first, we're looking at how quickly we could run the system. So we have the green graph shows us particle size, and we have speeds from 100, 200, 300, up to 500 mils per minute. And you can see the particle size is basically controlled by the production speed. So at 100 mils per minute, we're about 150 nanometers, but we can take that size right down to 100 nanometers if we're running at 500 mils per minute. So the white dots on that graph are the PDI, and you can see they're always sitting around 0.2 or lower, which is really good. So that's generally a kind of cut off where we see we have a good product. So the blue graph below is encapsulation. So basically that's telling us how much RNA we have. And you can see across all the production speeds, it's very high. So we basically get high loading and control of particle size through the speed. So all of these were produced at low pressure, and we ran at an aqueous to ethanol ratio of 3 to 1. And as I say, we were running up to 500 mils per minute. And that's actually the stopping point for us, because that was the upper limit of the current pumps we are using. So the speed can go higher. It was just where our pumps were running at. So we knew we could run up at 500 mils per minute, so we decided to keep running with that. So we were running the system at that. We were then taking samples at time intervals, so 20, 40, 60, and 80. And basically that's a different volume. So basically you could see we were producing at 166 to 600 mils, 666 mils per minute. We were taking samples to see how robust the process was. So where are we getting the same product coming out as we were running the system? So if you're running it continuous, are you always getting the same product coming out? So again, the green bar shown here, we have zeta potential on the left and PDI on the right. And basically what we're showing here is we were getting a nice steady state in terms of particle size and polydispersity. So as the system was running, it was producing the same product throughout the sampling process that we took. So after 80 seconds, we had over 600 mils of product, all highly reproducible in that respect. And in terms of encapsulation efficiency, again, it was very reproducible, all sample time points that we took. So you can see it's about 97%, I think, shown here. So good reproducibility, good quality control within the production process. We then wanted to check we do have process control. So we've already shown that particle size was controlled by the speed of the system, but there's also various other process parameters that can be controlled through the mixing ratio and also the membrane pore size selected in the operating system. So again, our green graphs are showing us the size and PDI. And as we move from left to right, you can see we can control the particle size we choose by optimizing the membrane pore size and the mixing ratio. So we can have them 100 right up to about 130 nanometers in size, depending on what size of particles you want. Generally 100 is seen as good, but there hasn't been enough exploration yet into what is a key LMP size. So that's something worth considering as we move on. But you can see all these size ranges, we are getting high encapsulation. So size is not impacting on our loading. We're always getting above 95% loading with these systems. So that's great. So basically we have a robust system that can run fast speeds. We can easily sample throughout and get the same product out. And if we want to vary the size, we have in-process control using the system. Another thing to consider is, if we're running at high throughput, do we get high yield or are we losing product in the process? So we did run the system at max speed again, 500 mils per minute. Bearing in mind that's the max speed of our pumps, not the overall max speed that can be achieved with the system. And basically we checked our yield, so how much we got out of the system. So at the very beginning, when you run it, we put to waste a very small start. So when we're running at 500 mils per minute, we discarded the first 3 mils just to make sure we're at steady state. After that, the production speed's running, and you can see we ran it from 100, 200, 300, 500 mils per minute, and our yield, or our product coming out the other end, is always over 95%. So we're not losing mRNA and we're not losing lipid in this process. Both is important as both is highly expensive for us, so we didn't want to lose that. So if you look at that, if we were running at 500 mils per minute, in terms of a Moderna volume of 0.3 mils per dose, we're basically producing 160 doses per minute running this system with a high yield recovery. So we're very excited to look at that, and it's definitely got very good potential at making these LMPs, as has been shown here. I'm now going to pass over to Sam, who's going to tell us a bit more about how the actual system works, because we were just actually using it in our hands. Over to you.
Brilliant. Thank you very much, Yvonne. I will take control. Yeah, thank you very much for that. Yeah, so a great overview of the initial work that we've done with Yvonne at the University of Strathclyde and the kind of current state of the art for these RNA-carrying lipid nanoparticles. What I'm going to talk through now is how we can use the Micropore Technologies platform to help you accelerate your development of injectable particles with a focus on lipid nanoparticles and liposomes. All of this with the aim of improving the control and reliability of your product, helping you to scale production with easy-to-use equipment with the end goal being reducing your development times and manufacturing costs for making these products, and show how we can work with you at all the stages of the development process, from discovery through preclinical, clinical, and then onto your manufacturing as well. First, a bit of background on where Micropore have come from and why we decided we wanted to get involved with this space with lipid nanoparticles and liposome production. As Tim said at the start, our background is in high throughput production of various different particle and emulsion systems. On the left, you can see some examples of some particles and emulsions that we produced before, things from PLGA encapsulations for long-acting injectables, interfacial polymerizations for other consumer applications, and simple topical emulsions, you can see shown in the top right there. All of these produced with our patented Crossflow technology, one of the units which is shown on the right, which is very similar to the unit that went to Yvonne for the testing that she's been carrying out for us. The way that this unit works, we essentially have one flow cell holder, as you can see here, with 3 process connections on it. One for what we call the disperse phase, one for the continuous phase, and then a product outlet line. The disperse phase inlet and continuous phase inlet simply connected to 2 pumps to feed the liquids, nothing else required there. And then inside the device, we use the tubular membrane, as Yvonne mentioned. These are stainless steel membranes with precision engineered pores. We control the size of the pores, the location, and the distance between them to give us excellent control over the membrane properties, but also excellent robustness and cleanability. Because the pores are straight through, they're very easy to clean using things like ultrasonics, and because we have control over the formation process, we can easily scale the throughput. The pore structure also gives us that very low pressure drop, which is great for this sort of application, especially if you're working with sensitive materials such as RNA, where you don't want to expose it to excessive shear. So as you can see, as the disperse phase is injected into the top of the device, passes through the membrane pores, and in our typical applications would form droplets in the center as the flow of the continuous phase detaches it. What this means is that the device itself has no moving parts. All of the energy required for the process comes just from the flow of the liquids. So just a pure stainless steel and PTFE construction here, PTFE for the seals, stainless steel for the holder in the membrane itself, gives you a very robust, scalable manufacturing process. This robust device lends itself to excellent control. So we've shown that for our particle formation applications, we can have excellent control over the droplet size, and we have a model for understanding this. This gives us an excellent level of predictability that allows us to easily scale customer formulations from the small scale in development all the way through to manufacturing. You can see here that we have reproducible, understandable kinetics for the droplet formation process. Reflected here by 3 different materials that we produce with the device by just changing the conditions as you saw on the curve there. So by changing conditions, we can alter the size of the materials. Additionally, using the technology, we've shown that we can easily scale the throughputs that we can get by adjusting properties of the membranes. So going from 5 milliliters per minute up to 80 milliliters per minute without changing any of our droplet sizes. Excellent control demonstrated here, again, made possible through the structure of the membranes that we've developed and the construction of the rest of the device and the control over the flow rates. We currently deliver this to a variety of different customers in the form of 3 different devices, the latest of which we're just launching now, which is the AXF Mini on the left, capable of operating as low as 1 milliliter per minute, all the way through to our AXF N, configured with 7 tubes as an AXF 7, to deliver up to 1,500 liters per hour of product. So we've been able to develop this full range of Crossflow technology to serve the entire industry, going from very early stage development through to the final stages of production, whether you're wanting to work with milliliters of material or wanting to work with tons of material. We have solutions that can satisfy that entire range. And as we mentioned, our previous experience has been doing this within the particulates and emulsion space, to be looking on the micro and above scale. The work that we'd initially got involved with Yvonne was to see how we could get involved with the mixing process for lipid nanoparticles. Our device at its core is obviously those 2 liquid streams being mixed in a controlled manner through a membrane. That's obviously not wholly dissimilar to the process that we're seeing for the formation of these mRNA-loaded lipid nanoparticles, where we're mixing the lipid mixture in an organic solvent with the aqueous buffer containing the mRNA. And what we wanted to do is to demonstrate and understand whether the same level of control we had for our droplet formation technology was appropriate for forming these particles as well. So the conventional methods for mixing that are used for forming these sort of particles obviously include microfluidic mixers, T-junctions, and impinging jet mixers. Unfortunately, these devices can have somewhat limited throughput. Typically capping out around 200 milliliters per minute is reasonably common for some of them. And so what we wanted to do, as I said, is apply our long experience in our high throughput, low pressure membrane emulsification to this mixing application to see if we could provide a system that allowed you to use the same mixing, the same formation process all the way through from development through to your final manufacturing and to give you a better product in the end. So as illustrated here, we're still using exactly the same device with the same membrane structure in the center, but rather than forming droplets, we have this controlled micromixing occurring. This is very similar to a jet in crossflow type mixing, where essentially by controlling the velocity and the density of the phases, we can control the mixing. Controlling the velocity is one of the key parameters that we control for our droplet formation process. So the modifications that we can make in terms of changing flow rates and changing the behavior within the device, as well as changing the membrane properties, we believe should lend themselves well to forming lipid nanoparticles with this equipment. After doing the initial work with Yvonne at Strathclyde, we were obviously very encouraged by the great results they were able to get, achieving 500 milliliters per minute production rates. What we wanted to do is demonstrate in-house what techniques we could use to control that lipid nanoparticle size and demonstrate that same level of control as we have for droplet formation. So as you can see here, there are two charts that we're presenting here. So on the left is the effect of pore size on the Z-average particle size and the polydispersity index. You can see that by adjusting the pore size, we're able to show control over the size of the lipid nanoparticles that we're making. You can see as we change from a 40 micron pore, we're forming at 99 nanometers, and as we go down to 10 micron, we've reduced that down to 89 nanometers. These are the exact same parameters that we can use to control the droplet formations. This shows excellent agreement for our control over the process there. The other changes that we make are to the annulus size within the membrane itself. By adjusting the annulus size, we're effectively increasing the velocity for a given flow rate. And by adjusting this is an extra parameter that we have for control within the device. As you can see here, as we reduce the height of the annulus, we increase the velocity of the materials passing through. So at the larger annulus size, forming materials at 115 nanometers, and then at the smallest annulus size, forming materials at 92 nanometers. So again, showing that excellent control over the size of the nanoparticles by changing not the formulation or the total flow rate ratio, but by just changing properties within the device itself. The next parameter that we wanted to explore was the scalability for the system. So the initial work that had been done at Strathclyde was using a series of gear pumps that we used. So one gear pump on the continuous phase, one gear pump on the dispersed phase connected to the device to mix the materials. We then expanded that in-house using two peristaltic pumps. This is obviously fantastic to integrate into a manufacturing environment because peristaltic pumps are obviously an option for contactless flow, and that will allow you to develop a fully aseptic flow path. So the full equipment itself, being stainless steel and PTFE, is suitable for full aseptic operation, so it can be sterilized in place, and we can help develop clean-in-place strategies as well. But as you can see here, we expanded that total flow rate range up to 1.2 liters per minute with one of our single AXF modules, and continued to exhibit the same good control over the size and the same very low PDI values that you can see here. This is done with the same, well, a very similar lipid formulation to what was worked on at Strathclyde, as you can see there. So very relevant to what's currently being worked on for a lot of the vaccine formulations. This obviously works the same way across all of our different AXF devices. So from the Mini through to the AXF1 through to the AXF7, where we're scaling out by adding multiple membrane tubes. This obviously can help to provide confidence that any initial work done at low flow rates can then be scaled up onto higher flow rates and improve throughput that way. Additionally, because we have that continuous production process, and we've now demonstrated that we have control by adjusting things like the flow rates and the velocities, this can integrate well with any sort of inline DLS to give you excellent process analytical technologies to give you control over your process as you're running it. You continually monitor sizes of materials as they're being formed and use that to regulate the process. This is, of course, data on the larger side of the flow rates. Using our new AXF Mini that we're officially now launching, we've also been able to work with this at much lower flow rates. So as you can see here, using our AXF Mini, able to work at total flow rates as low as 4 milliliters per minute, while still forming materials around 120 nanometers in this case, but still with PDIs less than 0.2. Typically, the target set by the FDA is less than 0.3, prefer less than 0.2, so we're coming in exactly at where things would be expected. Worth reiterating again, this is with that relevant formulation. So this is very similar to what is being used in the actual vaccine development. Obviously, different formulations can give different PDIs. Simpler formulations can typically give smaller sizes. But this is with a relevant system, and we're getting relevant PDIs with a simple aseptic process that is fully scalable from this small scale all the way up to liters per minute. You can see there some statistics for the AXF Mini on this side as well, suitable for working with around milliliters of material. So absolutely fantastic for early stages of development. As you can see here, so obviously the first stage for a lot of first experiments is just to produce 1 to 2 milliliters of product. Obviously you have budget material costs. A lot of the lipids that are being used for development are very expensive. You want to have things to be extremely repeatable. You want to make sure you're getting the same results every single time. Your device is easy to use as possible. This is the space that we hope our AXF Mini technology will be able to fill. So with its very low dispersed phase hold-up volumes of around 0.1 milliliters and very small working volumes, so you're able to work with dispersed phase low as 0.2, obviously just to cover that hold-up volume, and very quickly achieving steady state operation. Device here still functions in the same way as all of our other Crossflow technologies. One port for connecting your dispersed phase, one port for the continuous phase. But in this case, rather than using a gear or a peristaltic pump, simply connected to 2 syringe pumps. So you're allowed to you can easily and quickly change your conditions on the fly, produce small different size samples. Change conditions, change sizes, collect a different batch, change conditions, change your outcome size, collect a different batch of material. So you can very quickly and easily develop using this AXF Mini technology because of that continuous crossflow technology that we're using across all the devices. As seen here, still just simple construction, all 316L stainless steel with some PTFE sealing O-rings in there. So absolutely no consumables because the membranes are stainless steel, they're robust, reusable, and very easy to clean. Going ahead into our development, we're now wanting to produce slightly larger scales of product, around 10 to 20 milliliters. For this, we do not need to change from the AXF Mini device. Now, rather than testing a range of different conditions in our production run, we can now simply produce the batch of material that we need. So we see generating slightly larger volumes of material, but using exactly the same technology. No need to transfer to a different device. As you can see here, being able to demonstrate excellent batch repeatability with the Mini here, hitting the same size repeatedly from multiple different measurements and multiple different runs. As we move into the larger scale, producing larger quantities of product, in this case may be applicable to stick with our AXF Mini device or potentially move to our AXF1 to achieve those larger flow rates that have been discussed in the Strathclyde work from 100 to 500 or indeed up to a litre per minute of material. The good thing about running a continuous process is that even if you are running at a litre per minute, you can simply run for a few seconds at that rate and produce obviously smaller batch sizes. Because all of the equipment has very low hold-up volumes and equilibrates very quickly, it's suitable for that sort of operation as well. Yep, so as you can see here, with the devices have all been designed to be suitable for GMP production. So of course, all electropolished stainless steel. We can provide material certificates for any of the parts that are provided, drawing packages, and any level of support that you need as well to assist in that validation as you continue to move through the regulatory process to getting your product to market. And then finally, moving to the manufacturing scale. At this stage, you don't need to be looking at changing your devices. Essentially, a lot of the troubles associated with tech transfer, if you're moving to different kit, are completely eliminated. All of the learning that you've been doing using the AXF Mini or even the AXF1 at the earlier stages is completely translatable because you're still using the same membrane crossflow technology that we do have that modeled understanding for. All of the data that you've generated at your early stages is relevant to go on to your later stages. As you're developing your process to integrate this, we can continue to work with you and integrate things like PAT control. We can integrate inline dilution, options for TFF steps for the total process as well. So we can continue to work with you to develop a fully specialized system to produce exactly your product and give you the level of control that you need as well. So as you can see here, the full range of equipment covering the full range of flow rates that we can, from as little as 1 milliliter per minute with the AXF Mini all the way up to 1500 liters per hour with the AXF-N systems configured with 7 tubes. All the devices, very low holdup for the quantity of materials that they're running, both in the disperse phase and the continuous phase. Allows you run with very small volumes of disperse phase in total, and running very small volumes to actually achieve steady state. So if you want to run at a higher rate but only produce a smaller quantity of material, that is possible with the equipment. As we mentioned, all constructed out of 316L stainless steel with some PTFE O-rings in there, all electropolished, no moving parts whatsoever. So obviously excellent for your validation. So as I said, our goal has been to provide a technology platform that can help accelerate and simplify the development of any injectable products. This presentation focusing on what we can do for lipid nanoparticles, with the hope that we can reduce your development timelines and manufacturing costs so you can improve your throughputs and reduce your processing times. All of that possible with the 3 devices that I've talked about today. So from the AXF Mini at your early stage, going through discovery and preclinical, moving to the AXF-1 when you're ready to, using all of that same data and control that you've already generated and still being relevant as you continue through the process, and then potentially finally moving to one of our AXF-N units at the final stages of manufacturing if you need the absolute highest throughputs possible. So in summary, this is our continuous cross-flow micromixing process. It provides highly reproducible results for formulating any sort of particulates. It can give you precision control over lipid nanoparticle size. Currently we've demonstrated from 60 nanometers to 250 nanometers, so this is very formulation dependent. The system is simple, robust, and very stable in operation. We're running with a very low pressure, low energy process that can help preserve your fragile materials. Typical operating pressures less than 2 bar. Also very low shear inside the device. As I said, very low pressures, and for the flow rates that we're running at, no large amount of shear generated because the membrane design lends itself to low shear as well. All the devices have been designed around very low hold-up volumes, so you're not using excessive material when you're running your testing. We can provide these with a completely aseptic flow path. If we use things like peristaltic pumps, so you can have a contactless flow path there, that means that you can make these materials without needing to carry out sterile filtration. So for those who are interested in targeting larger sizes for nanomaterials, larger than 150 nanometers, getting close to that 0.2 microns there, that is possible with the system because it is suitable for fully aseptic operation. All stainless steel and PTFE, so obviously suitable for things like seam-in-place sterilization. It can also be disassembled simply and cleaned out of place. And as Yvonne mentioned as well, we can quickly and easily get anybody set up with the equipment, whether that's in your lab or in an R&D environment, or through some trials that we run in partnership. We've worked with a variety of customers around the world. Currently, 11 of our systems are in use around the world that are in clinical trials, many more in the development stages before that. But some of the people that we've worked with that we can talk about on the side of your screen there, on our particulate development technologies. Thank you very much to Yvonne for her earlier part of the presentation. Thank you to all of you for listening. I hope what you've seen today has been interesting. If you do have any questions, please do post them in the chat. Yvonne and I will be here to answer any questions that you might have. If not, feel free to get in touch with us afterwards.
Great. Thank you, Sam, and thanks, Yvonne. It was a really great talk. And like Sam just mentioned, we do have time for some Q&A. We've had some come in here, but if you do have a question for either of the speakers and you didn't get it in, now's the time you want to go ahead and type it into that box there, and we'll get through as many as we can. So I'm going to throw these out there, Sam and Yvonne, and you guys can decide. Take some, and we'll see what. Kick things off here. This attendee asks, can DMSO be used as a co-solvent for the LNP versus ethanol?
From an equipment perspective, absolutely fine. Yvonne, I don't know if you want to weigh in from lipid solubilities or anything.
No, that's a good question. So we have used DMSO to dissolve some lipids because not everything dissolves in ethanol, as we know. So yes, it does work. In previous experience, it tends to push the size up a little because it takes us longer to change that polarity. The ethanol, it changes quickly, so you get the small vesicles. With the DMSO, it takes you a bit longer to get that polarity change, so we tend to get slightly larger vesicles. That is another thing to consider depending on your solvent. And you can blend them, so you can blend ethanol and DMSO if you want, and actually you get intermediate particle sizes as well. So it's quite a cool thing to blend your solvents.
Yeah, an extra degree of control outside of just the equipment is obviously formulation controls. Yeah, all possible with the equipment.
Okay, thanks, guys. Let's see here. How large a diameter NP can you produce?
Currently, the largest we've produced is about 250 nanometers. Though kind of as Yvonne was just indicating, we've been predominantly working with ethanol when we've done that. So it's possible that with different formulations, larger sizes will be possible. You know, a lot of this work has been carried out here over the course of the past 6 months. As I indicated, our background is in the particle formation, so injectable particles and emulsions. So we're really sort of starting to develop more around the liposomal lipid nanoparticle capabilities. I'm not sure, Yvonne, if you've produced any larger materials than the 250?
No, I think we've been slightly in a smaller size range than that. So yeah, I think we were about 80 to 150 was the control sizing we had. So
yeah, that seems to be the target we see a lot.
Okay, thanks, guys. This attendee would like to know, can the pore size of the system be customized, or are there standard sizes?
We have a range of standard sizes, but we can also do custom sizes depending on the application as well. So the way that we'll typically work with a customer is we like to run some small in-house trials first, so we can determine the optimal equipment configuration. So we can run some small in-house trials, potentially produce some small batches. And then if we get to providing a full system, we can provide a customized full system along with a process guarantee as well. And as part of that, that could be a custom membrane configuration. Typical sizes, you know, we typically go up in denominations of 5 microns, for example, but if we found the application required, you know, 7.5 microns is the optimal size, as we saw from that graph of controlling lipid nanoparticle size, then that's absolutely an option and something that we would be able to produce for you.
Okay, thanks, Sam. Let's see here, moving on. What is the maximum potential mRNA concentration?
Well, I can come in and say we've started, so we've been running at 20 microgram per ml, and then basically we would concentrate that to 40 because there is we have to remove the ethanol, if you remember, because of this process. So we actually tend to make it more dilute so that we can basically suck out the ethanol with TFF. You can go much higher. Basically, we haven't gone much higher because there's no need to concentrate it up more for our studies, and obviously the cost of mRNA can be quite prohibitive. But easily we can work at the concentrations required, and as I say, most manufacturing processes, the final concentration is controlled through the tangential flow filtration step process. Sam, I don't know if you've got anything else you want to add?
No, yeah, covers it perfectly. I was just going to say it ends up being very formulation dependent on what you're doing for your lipids and then how your downstream processes look as well. Yeah, no equipment limitations on that, certainly. No,
definitely not. I think the general limit is actually the solubility of your lipids in the ethanol. That's what we find is our key controlling parameter at the very start of the process. And then that kind of dictates what you get out. It's not how much the system can handle.
Right.
All right, great. All right, let's move on here. This attendee asks, are the membranes exchangeable, or does a new system have to be built for additional pore sizes?
Yeah, so the membranes are exchangeable. So you can easily disassemble the device and remove the membrane. So essentially 2 ends holding the seals that seal the membrane inside. So the membrane tube can be removed for cleaning out a place, or swapping out for different membranes. And Yvonne, the system that we sent you came with a couple of different membranes I think you tested.
Yeah, that's right. So we were just swapping them in and out in that process development. So the graphs we had where we're showing the different sizes, we were basically varying the inserts and taking them in and out, and just a standard run-through cleaning process before we swapped them out. But yeah, very easy to do.
All right, great. Let's see here. Why are the membrane pores so much larger than the formed particles?
Okay, yeah, so obviously from the mixing process, so obviously mixing the lipids in the ethanol, as the ethanol obviously becomes mixed into the buffer phase, that's just lipids on their own, as Yvonne outlined, they obviously sort of self-assemble into these vesicles. So it's not actually, whilst the pore size does help to control the size of the vesicles, that's more because of the size of the stream of liquid that we're introducing, rather than the pore size directly. You know, it's 40 microns, so it should make a 40 micron particle, for example. It's just essentially providing an avenue for introducing a liquid in a controlled manner. So it's because of that precipitation that is causing lipid nanoparticles to form, rather than exactly the pore size controlling it. Hopefully that answers that.
Okay. Anything to add there, Yvonne, or does that cover it?
Yeah, no, exactly. It's all about the mixing process, so it's not an extrusion process as was outlined. So it is generate the mixing, and the mixing causes the nanoprecipitation. That's how we get the LNPs.
Right. Okay. Okay, moving on. What is the life of the membrane? How many cycles or batches?
Yeah, I can handle that one. So because, as I said, so 316L stainless membranes, and the pores are precision engineered to essentially be straight through channels in that, gives them an extremely long lifetime. We have some membranes that we're still running here as part of our development unit that were actually around when we first started prototyping the device back in 2016. They're still in regular use here. Provided you're using appropriate cleaning cycles, absolutely unlimited lifetimes pretty much, which is great for you as an end user. Provided you're not doing anything that could potentially degrade the membrane surface, so exposing it to particularly strong acids or anything that's going to interact with the stainless steel, or putting anything through that's larger than the pore sizes, or that could essentially block those pores, because as much as they are straight through channels that are designed to block, not block as much as possible, if you try and put, a 20 micron particle through a 10 micron pore, it's not going to work. Fortunately, though, you can clean that back out, so you can reverse pressurize the membranes and force material back out of the pores. So even in those cases, they are very robust. So I'd say the lifetime is, with proper care, near infinite.
All right, thanks, Sam. Let's see, we're going to take one or two more questions. This attendee would like to know how to clean the membrane, through IPA or through any other method. I guess what they're asking, what's the best method?
Absolutely. Yvonne, do you want to talk through the method that you used?
Yeah. So we just do a normal ethanol flush, to be honest. So we tend to do a 70% ethanol flush, and then just a standard water or buffer flush, depending on what we're going to do with that. But that's our standard protocol. If you're going to work with RNA, remember you might want to decontaminate it with RNA ZAP, for example. We would flush it through with that as well, just to make sure you are free of enzymes. But standard ethanol flush is what we do, just out of process. But most of the times, I think just a buffer flush would actually do the trick.
Yep, so either you can obviously clean it in place like that, forcing material through the pores, or clean out a place using ultrasonics. That's what we'll do typically for the sort of particulate-based applications. If we want to try and free up any material that could be in the pores, the ultrasonication can help with that. But obviously the clean in place flushes are obviously an optimal way of working with the membranes.
All right, great. I think we're going to take two more here. What is TMP, transmembrane pressure? I guess they're asking what that stands for.
Yes, so TMP would be transmembrane pressure. Okay. So that's obviously the pressure required to force the liquid through the pores. In most cases, less than 2 bar, because we actually have very high open areas on the membranes themselves. And we also have a full modeled understanding for determining what that transmembrane pressure would be for trying to force liquid at a given flow rate through the membrane. And as I said, in most applications, it will be less than 2 bar. in fact, actually because of the solvents that are typically used and the concentration of lipids for a lot of these applications, more commonly less than 1 bar in most cases here.
Okay, thanks. We're going to take one more question before wrapping things up, and this attendee would like to know about how to protect mRNA from not denaturing during the manufacturing process when organic solvents are also present.
Yeah, I can maybe pick that one up first. So all the processes that are making the LNPs do have some organic solvent, and ethanol is generally the chosen one because it's easier to handle manufacture. So there's been various studies showing that the RNA remains intact through the ethanol because it can withstand that. That's not a problem. And also it's a rapid mixing process, so the time it's actually exposed to the ethanol is quite small. To be fair, depending how quickly, I mean, if you imagine we're running at 500 mils per minute, but even at the slow ones, the exposure to the neat organic solvent is very minimal. But that is all the processes that use the same method.
Okay, great. Thanks, Yvonne. Yep, so that's going to conclude the Q&A portion of the session and bring us to the end of a really terrific talk. If we didn't get to your question, Sam and Yvonne have your details and will respond to you directly. And we also had a couple of you ask out there if the presentation will be available for download. And for those of you that did ask directly, Sam and Yvonne do have details and we'll get back to you directly. And if you are interested, you can email Sam at sam.trotter@microporetech.com. Again, that's sam.trotter@microporetech.com. And also feel free to reach out to us at Contract Pharma, and we can put you in touch as well if you did want a copy of that today's presentation. So thank you again, Yvonne and Sam, for a really terrific talk. Thanks to today's sponsor, Micropore Technologies. Be sure to check them out at microporetech.com. And thank you, everyone in the audience, for your attendance and participation today. As I mentioned at the top of the hour, this session will be archived for 1 year on our website, contractpharma.com. Thanks very much for joining us, and have a great rest of your day, everybody. Take care.